As a labor economist, and I know there are a lot of labor economists out there, you'll appreciate this application, but there are a lot of other things in life that look like labor, employment, and unemployment spells. So I hope that's why you're all here and also numerous and attentive. So Roger, the floor is yours.
Okay, so as Michael said, I'm going to try to survey some recent and a bit of not so recent work on duration models and how quantile regression might be useful in looking at them. I'll say more about this picture a little later. It doesn't look like a very economic picture, I'm afraid, but and it isn't, but I have some way to connect it to economics, or maybe a couple of ways to connect it to economics. Starting point of this is really related to this distributional regression stuff that I talked about earlier in the morning. Except that now I want to think about this first equation as a duration model in which t is an event time. And for the moment, I guess I want you to think about uncensored event time, so that we actually are observing all the event times. We'll talk about censoring a bit later. We have a vector of covariates, an unknown parameter. We have same kind of thing, IID error for the moment, just to keep things quite simple. But the whole point, of course, is that we don't really believe in the IID error assumption, and we're going to try to get rid of it at some point. So just as in the morning distributional regression stuff, the idea is to transform such models so that we have something simple that we can estimate. In the morning thing, we were trying to estimate a binary response model, and one can still do that. There's this nice paper by Dave Donahoe and Miriam Gasco, it's Shel Doxson and Miriam Gasco, that connects these binary response models and the survival analysis models. And the best known of the survival analysis models is obviously the Cox proportional hazard model. And in the Cox model, you get this complementary log-log expression that we basically had this morning, although maybe it's not entirely recognizable the second time around. But the idea is that in the morning's discussion, we were transforming some probability, and then we got a kind of time-dependent intercept. So that was the thing that was indexed by y in the morning, but now it's indexed by t because we're doing durations rather than just an arbitrary response. And then we have these additive linear predictor kind of covariate effects, which are x transpose beta. But the nice thing about this is that we don't have to do the binary response model. We can substitute into the survival function the random event time, capital T, and then we have what you would, I guess, want to call a general transformation model, except that in this case, log capital lambda naught is just the specific transformation that we need to use in order to deal with the Cox model. And in the Cox model, we would have this kind of regression equation at the bottom with error that's IID and having this kind of type 2 extreme type. I always get my type 1s and type 2s extreme value theory messed up, but I think this is type 1, I guess. You can do the same thing with the so-called Bennett model, which is a kind of variant of the Cox model that is doing logistic kinds of things. Now you've got log odds as something that you can write as an IID error model, and again you get some kind of complicated transformation equals linear predictor plus IID error. And so for each of these particular choices of the error in the original model, there's a kind of way to go back to a binary response model, or a way to do a survival model that really is representable as linear predictor plus IIDR. Of course, the trick is to know what the right link function is, what the right transformation function is. And that's the thing that we're going to try to get away from to a certain degree by doing the quantile regression. That is, we may not really know what the right transformation is, and we'd like to avoid trying to have to estimate it directly. The third example, which is even more straightforward and maybe more familiar, is to just forget about the fact that we're going to make some kind of distributional assumption on the U's, just instead say, what's the simplest plausible distribution that we could, or possible transformation that we could use on T that would keep the positive responses mapped into the whole real line. That seems to be logarithm. And so now we look at the consequences of assuming that, and we get this, simple accelerated failure time model in which time is either speeded up or slowed down in accordance with this factor e to the minus x beta. And now we get this, the hazard function from the corresponding model. And so this accelerated failure time model is a kind of standard thing that's used quite a lot in the empirical literature. And it has very simple interpretation that is kind of convenient. So these are three of the leading standard survival analysis models. And now the question is, can they be adapted to quantile regression methods? And can we then get away from this IID error assumption? Can we do something other than assume that the covariates are just shifting the location of the term involving the linear predictor, and the intercept is just following along as a kind of scalar shift effect? It seems quite plausible in a lot of settings that we need something more general than this IID error assumption. So to motivate this, I'd like to talk a little bit about an application that I was involved in several years ago. The data actually comes from experiments that were conducted in Mexico by a group of people that I guess were at that time centered at University of California, Davis. James Carey was the kind of lead author in this. And the idea is that we're going to study lifetimes of these medflies. So medfly is a pest in Southern California, particularly in citrus orchards. And they raise these flies in Mexico on a very large scale and then release them into the wild in California. And these flies are raised so they can't reproduce, and the result is that it confuses the native population of medflies. And so this is a quite, at least 20 years ago, this was quite a major activity, but there were these large factories in Mexico that raised these flies on a large scale, and Carey and his colleagues convinced funding agencies to allow a large number of these flies to be raised in Mexico for their experimental purposes. And for that purpose, they raised 1.2 million of them and then followed their life history in cages in this factory in southern Mexico. And when Olga Gelling, who was a student in the stat department at that time, came to me and said she had been at a conference in Rostock and that she'd heard about this data set, and she started telling me about it and said, we have 1.2 million observations on lifetimes without any censoring. I thought, gee, this is really too good to pass up. So Carey was kind enough to allow us to use the data. And it turned out that there were certain remaining issues that were a bit controversial with the initial analysis, and I'd like to just briefly talk about them because they're kind of interesting, I think. And it does sort of illustrate what can be done with these methods in duration context. So they're basically raising these flies, and they're all in some sense born at the same time. And each day somebody comes around and sweeps all of the dead flies out of the cage, and then somebody then meticulously looks to see whether each fly was male or female, and they count these things at each day. And so we have lifetimes of these flies by sex, by cage, by various things, for a period of about almost a year. So the result of these experiments was quite a dramatic change in how people thought about mortality and survival distributions in lower animals. I think that it's fair to say that the standard biological view of aging, to a certain extent, was dramatically influenced by this one study, and as a result there have been a lot of subsequent studies on lower animals and some higher animals in an effort to better understand a variety of the results of this study. The most striking conclusion, I guess, was the apparent decline in the mortality rate at advanced ages for these flies. And this contradicted a really strong preconception of the biology that aging was simply a prolonged decline into senescence. This is a phrase that you see over and over in this literature. But in fact, with these flies, it appeared that once you got to a certain age, your mortality risk actually improved for a little while at least, and then it wasn't so clear what happened. But other conclusions were also important. That is, one of the things that they found was that the tail of the distribution was much longer than they originally expected. And there was this general view in the biology literature that each species had a kind of fixed lifespan that couldn't be extended in any way. And here we really see some kind of statistical view that's quite different than that. That is, that there's really a tail to the distribution, and it's not really a meaningful thing to say that there's a fixed lifetime beyond which these flies actually live. There's just a kind of gradual decline in the tail. And finally, there's a kind of interesting gender crossover, which I'll talk about a bit. So here's a plot that's more or less a reconstruction of ours from a plot that originally appeared in the first Carey paper in Science. So you have lifetimes in days on the horizontal axis, and the mortality rate on the vertical axis. And what you see is that for both male and female flies, that after about 60 days, the mortality rate declines. And roughly speaking, a fly day, or at least a medfly day, is something like a human year. So for people like me, this is kind of encouraging news, I would say. It suggests that, for reasons which are not so clear, once you get to 60 as a medfly, your life chances actually improve a little bit over the next period, up to about maybe 80 days, and then the data becomes a bit sparse, and it's very hard to know exactly what the precision of these curves should be. But the original work and the work that I'm going to really present doesn't really have too much to say about the precision, certainly in the far outer tail. But you can see that There's this interesting gender crossover too at about 20 days. Females have higher mortality risk up to about 20 days, but then they have lower mortality risk than males after 20 days for a while until you get into this later period where they're really indistinguishable in terms of mortality rate or hazard. So to compare this with humans, which is a bit inevitable, the people who work on Drosophila and also on this medfly stuff always like to say that after you even begin to think about this for a little while, you begin to think that flies are little people with wings. You can kind of see what the mortality table would look like. So we have roughly 1.2 million flies on the left side, and this is kind of illustrative of the long tail of the distribution. Only 1% of the flies are actually left after 50 days, but then you have flies living to 146. And we don't really have good evidence on human populations beyond about 115 or something like that. But you can see that it's very hard to sort of say, well, there is a kind of fixed limit for the medfly tail behavior. In fact, some of these 12 flies that actually managed to get past 146 lived over 200 days. Whereas, if you look at the kind of, originally I had the idea was that this human population was the same size, so the last column on the right is supposed to be the decline in population for this population of this size of 1.2 million. But we still, the tail behavior is actually much thinner, but we still don't really know very much about, what the feasible extent of the human lifetime is. And James Vaupel, who I think is one of the more prominent demographers in the world at the moment, and who is, I think, director of the Max Planck Institute in Rostock, likes to say that girls born, now he's saying actually, I think, all children born, but I think it's more convincing to say that girls born in Northern Europe after the year 2000 have a median lifetime of 100 years. And it's kind of a remarkable thing when you think about it, but it's also a little scary if you're responsible for forecasting mortality tables for Social Security or something like that. So one of my kind of angles on this when I'm presenting to economists is to claim that this has some relevance to saving Social Security, or suggesting that we need to worry more about it, saving Social Security. Olga and I wanted to look at a regression version of this because all of the original work had been done with this very simple kind of life table analysis that is something that you could have imagined doing based on standard actuarial methods from the 19th century. It was all based on kind of univariate kinds of things in which we just could do a single survival distribution, maybe a single survival distribution stratified by males and females or something like that. But there was quite a lot of controversy after the science paper was published about the possibility that high initial density of flies in particular cages reduced life chances. And then as flies died off, flies were happier because there were fewer competing flies, and they should be therefore living longer. So we were interested in pupa size as one possible source of variation. Gender was an important thing because we were interested in this crossover effect. Initial cage density was a third thing, and then as a kind of, almost as a joke, we were able, because eventually we knew the sex of all the flies, to calculate the proportion of males in each one of the cages. So at each quantile we wanted to estimate a model like this with particular attention to what the upper tail of the distribution looked like. So here's a kind of standard quantile regression coefficient plot. In each panel you see the coefficient estimate and a confidence band for it. You also see in these red things the ordinary least squares estimate of the corresponding accelerated failure time model. So if we focus for a moment on the intercept, which is this the upper left panel, that's what the effect is for male flies with average cage density and average percent males and average size. And so that's the quantile function of that subset of observations quantile function. So, and then the second plot is the gender effect, and so there's for females there's this negative effect until you get up to about 0.95 or something like that, and then there's a crossover. The size effect of this is the size of the initial pupa that was introduced into the cages, is basically giving no effect whatsoever. So it doesn't seem to be really relevant in the analysis. But quite interestingly, from the point of view of all of the criticism of the original study, the density effect is quite constant and positive, meaning that being in a cage with more flies actually had a positive or an improving effect on mortality prospects, longer survival for more densely populated cages, which was exactly the opposite of what was conjectured by the critics of the original study. And then our kind of purely speculative last covariate, percent male, also has positive influence on survival prospects. And if you try to differentiate this by having an interaction between gender and male effect, it's true that for both males and females, there's a positive effect of having more males in the cage, which is completely a mysterious finding, I would say, from kind of all conceivable points of view.
No reason to have males, period.
Yeah, and, males are just good. I mean, and who could imagine why is the real question. Okay, so this is, I guess maybe there are a couple of other points that I would like to make about this. If you look at the density effect, what you see is a fairly constant effect over all of the quantiles. And so you're really not making a big simplification of the model to say that there's a location shift effect for density. And even for the percent male effect, it's quite constant. And so maybe the Cox model version of the fit, which is this red line, is saying something a bit different than what the quantile regression results are saying, but it's not an enormous difference. And you might be willing to say, okay, we're willing to make a location shift assumption for those two covariates. But if you look at the gender effect, it's clear that the Cox model is really giving something quite different than the quantile regression effect. And the thing about these crossover effects that's really, I think, important is that The Cox model and related models, I mean, all of these previous models that we talked about, accelerated failure time, the Bennett model, all of the standard IID error kind of survival models are assuming that covariate effects have to have a single sign, and they affect the hazard function or the proportional odds or whatever it is in some way that rescales those features of the distribution in a way that forces the sign to be of one, either positive or negative, but uniformly positive or uniformly negative over the whole distribution. Whereas in the tail here, it's clear that we're getting something in which the effect of this covariate is really different in the upper tail than it is in the lower tail. And the thing that I guess I would like to emphasize about that is just that certain modeling assumptions, even maybe without really intending them to, can have the effect of kind of blinding you to certain kinds of effects that are really there in the data. And I think this is an example of that. So the difficulty, of course, with trying to convince anybody that quantile regression is relevant for duration models based on this MedFly study, however interesting you might think it is that there's no treatment for censoring. And so it's really necessary to provide some kind of treatment of censoring if you're going to try to have any kind of impact on how duration analysis is done. And the MedFly data was particularly nice in this respect since it didn't have any censoring. But fortunately, other people have done very interesting things lately with quantile regression in censored situations. And so I'm going to talk just a little bit about three of these approaches. So the first one goes back to relatively early days of quantile regression, and I think has had an enormous impact on empirical work in some parts of labor economics and other areas in econometrics at least, and that's Jim Powell's work on fixed censoring. So the Powell original insight was just to take this monotonicity, the equivariance to monotonicity that Andrew Cheshire mentioned this morning, and observe that if we have a model like this in which instead of observing the original t's, we observe the minimum of t and some censoring times ci, then we can think of that function, min tc, as a monotone transformation of the original t. And so the equivariance property ensures that the conditional quantile functions of this new y after censoring is just the same transformation of the quantiles of the original model. So it's just min x beta ci. And so now we can try to estimate this new model by doing something that really looks like standard quantile regression, but isn't really standard quantile regression because there's this nonlinear in parameters specification of the response function. But the nice thing about it is that the optimization is very closely related to the original simplex approach to doing standard quantile regression. And Bernd Fitzenberger at Mannheim in his thesis at Stanford worked on computation of this, and there are quite good algorithms now for solving this problem at the So a second approach, which is a bit more connected to the standard biological literature on censored survival analysis, deals with what is usually called random censoring. So the Powell setup is fixed censoring, and the standard assumption is, in this setup of the model that Powell analyzed, that we observe the CIs for all observations. The simplest case is that CI is equal to a constant and it's just a threshold. It needn't be a fixed thing. It can be observation dependent, but we need to know all of the CIs. In the Portnoy setup, we have random censoring, and so we don't know the censoring distribution, but we are able to know only the CIs for the observations that are censored. So we have a censoring indicator that tells us this observation is censored, and therefore the event time that we see is actually the censored event time. And Portnoy, starting from the point of view not of this monotone equivariance thing, but from the point of view of trying to generalize the Kaplan-Meier estimator, invented a very clever way to reweight the observations in order to accommodate this kind of censoring. So without going into too much detail, the idea is something relatively simple, and actually goes back to a paper that Efron wrote when he was maybe still in graduate school, but it's in one of the Berkeley Symposium volumes from, I think, the late 60s. So the idea is that if when you look at observations in the simple one sample case with censored observations, what the Kaplan-Meier estimator is doing is saying, we don't know really how to distribute the mass for this censored observation over all of the region above the observed event time. I mean, in effect, we know that if the observation is censored at a certain time, it can't have had an event time below that, but it has to have had an event time above that, and we just don't know where that is. But from a quantile perspective, that just means that we can take the part of the mass of that observation and shift it to infinity. Well, from a computational aspect, not really infinity, because computers are not very happy with infinity, but some place that we know is going to be above where any of the other observations are. So in that case, if we do this as we move up through the data in somewhat the same way that the movie was trying to do this morning, we can define these weights that are given here, and then we solve a very simple quantile regression problem with those weights at each tau. And having moved up through the whole set of data, we can, in principle, then find the whole quantile regression process. And this provides a very simple algorithm in terms of optimization for what normally is thought of as the Kaplan-Meier estimator, or really the inverse of the Kaplan-Meier estimator. Kaplan-Meier is estimating the distribution function. This is estimating the quantiles of the Kaplan-Meier distribution. And this generalizes nicely to regression, as the usual thing does. We do the same thing with the splitting of the observations and define the weights in exactly the same way, and we have a generalization that provides a kind of quantile regression version of the Kaplan-Meier problem. This is a small advertisement, I guess, for doing this in R with my QuantReg package. But one enters data just in the usual way that one would do for a Cox model. This serve log time comma sensor is just the standard way that one enters the event time and the censoring indicator in order to form a dependent variable in these kinds of models. And then we can compare this with the fit from the Cox proportional hazard model, which I guess is done on the next slide. Yeah. So this is Portnoy's standard example in his original JASA paper. Again, I don't want to really dwell on this much, but this is data on a treatment program for drug addiction. But you see the red curve being the Cox model estimates, this blue band being the quantile regression estimates in their confidence band. And there are a number of cases where the two things look quite similar, but a number of other covariates have effects that really look very different than what the Cox model would be predicting. So now there's a third approach, which turns out to be quite closely linked to the Portnoy-Kaplan-Meier approach. So in the standard theory for survival analysis in the univariate case, we know that there's this kind of standard and kind of earlier approach due to Kaplan-Meier, and then there's a kind of recent or relatively recent revolution in analyzing methods of this type that is focused more on, less on estimating the survival distribution and more on estimating the cumulative hazard function. And the standard method for estimating cumulative hazard function is this Nelson-Allen method. And it turns out that because of the counting process formulation and because of the way that one can formulate the counting process formulation as a martingale, it provides a more direct way in some respects to doing asymptotics and deriving estimation procedures. So the idea is that more or less, as the Portnoy thing is saying, we have this model that says probability that log t is less than some linear predictor should be equal to tau. And then we have counting process NIT and this compensator function, which is the expectation of NIT at the moment before t. And so that's the thing that, if subtracted off, gives us the martingale process, MI. And this capital lambda is just the cumulative hazard function evaluated at the point t. And given the cumulative hazard function, we can then derive the distribution function or the survival function or whatever else we need. So the nice thing about the martingale thing is that it gives us an estimating equation. I mean, this is one of many situations in which quantile regression methods can sometimes be conveniently reformulated as just an estimating equation procedure. And fortunately, in this case, it's particularly simple. You look at the estimating equation formulation, which says that this particular function of the counting process minus the compensator equals 0, and you reformulate that a little bit, and that gives you a first order condition that then you could try to solve in beta, but it's a sufficiently simple first order condition that you can work backward to get an optimization problem. And so in effect, you're always at each stage in the process solving what turns out to be essentially, again, a weighted quantile regression problem. And the nice thing about the martingale is that it has the advantage that one can actually do asymptotics for it in a way that's quite a lot simpler than the Portnoy thing. In fact, Portnoy estimator, while it has a certain amount of the asymptotics nicely worked out, particularly consistency and rates of convergence, doesn't really have a nice, clean limiting normal theory, whereas this has a somewhat simpler version of the normal theory. But it turns out that the two estimators, not surprisingly, there's this kind of almost duality between Kaplan-Meier and Nelson-Allen. These two estimators, the Portnoy and the Peng and Wang estimator, behave very similarly in numerical experiments. And we should expect that they are asymptotically equivalent, because they are asymptotically equivalent, at least in the univariate case. This kind of raised some questions about relative efficiency of these procedures, and so it seemed interesting to look at, again, very simple cases in order to get some kind of clues about what was going on in the more general cases that didn't seem to be really that easily adapted to standard large sample asymptotics. So I'm comparing, I guess, 6 different estimators here. One of them is infeasible in the sense that I'm calculating, in every case, I'm trying to get the median of the survival distribution. And with censored data, we can't calculate the median of the as if we had all the data, but the first column is corresponding to the asymptotic, well, the finite sample variances and the asymptotic variance of the, actually, I guess, mean squared error of the median, as if we had all of the data. So this first median column is kind of infeasible because we're assuming that we know the actual survival times in all cases rather than having just the censored ones. Then we've got the Kaplan-Meier estimates and the Nelson-Allen estimates in this simple one-dimensional setting. And what we see is that both in the simulations and, of course, in the asymptotics, they actually behave very similarly. And then we have the Powell estimator. And the Powell estimator, it seems in small samples, is actually doing as well, or almost as well, as Kaplan-Meier and Nelson-Allen. But as the sample size gets larger, again, I'm scaling all of these things by root n in order to make them comparable with the asymptotics. In larger samples, Powell estimator looks like it's sacrificing about 10% efficiency. And this was kind of a mystery to me for quite a while, and it produced a lot of discussion at coffee conversations in Urbana with Steve Portnoy. And the mystery to me initially, which was, I mean, I think just pure naivete on my part, I kept saying to myself, well, the Powell estimator is using more information. I mean, it's assuming that we have CIs for every observation, and if we have CIs for every observation, we should be able to use those CIs in some intelligent way to actually produce something that's more efficient than what Kaplan-Meier would produce. Of course, that presumes that we're doing something really smart with these CIs. And it turns out that the CIs are not really relevant to the problem. I mean, if you read Jim Berger's book on the likelihood principle, there's a nice couple of paragraphs explaining in the one sample setting why CIs for the non-censored observations in such problems can be factored out of the likelihood and therefore don't contribute to the any kind of conceivable way to improve the efficiency of the estimator. So it appears that, contrary to the fact that they might have been used in a better way, that there is no way to use them in a better way, but if you use them at all in some kind of substantive way, it has the effect of deteriorating the performance of the original Kaplan-Meier or Nelson-Allen estimator. Then there are a couple of other estimators up here that I'm calling Lurgan's G hat and Lurgan's G. Sue Lurgan's has a paper in JASA which looks at some kind of propensity score weighted estimators of this type. I don't think she's very interested in this one sample case, but it's interesting to me to specialize her situation to this case. And so in the Lurgan's case, we can estimate the censoring distribution G by just looking at the empirical CDF of the censoring times, assuming, like Powell, that we know all the censoring times. Then we can do the propensity score weighting thing, and when we do that and we calculate the large sample theory for that, it turns out that it produces exactly the same behavior that the Powell estimator does. And if you look at the simulations, it performs a little bit worse than Powell for small samples, but it eventually looks quite a lot like Powell once you get up to sample sizes, 500 or 1,000. Then there's a kind of even more bizarre thing in which you might say, well, let's take the Lurgan's estimator, and instead of doing the propensity score based on the empirical CDF, you do the propensity score weighting by the true CDF. So surely the true CDF should be doing better than using the estimated censoring distribution. But as you can see here, it does much worse by almost, well, let's say 12% or something like that. And in small samples, it does even worse. And so there's this kind of funny ordering of the Lurgan's estimator. So I haven't mentioned this yet, but you could imagine instead of doing the true distribution or the empirical distribution that uses all of the censoring times, you do G hat as the Kaplan-Meier estimator for the propensity score. That's using less information than all of the censoring times, but that actually restores the efficiency. If you do that, you get back to the efficiency of the original Kaplan-Meier or Nelson-Allen thing. So there's this kind of reverse ordering in which the efficiency of the median estimator here is in some way made worse and worse by better and better estimates of the censoring distribution being used as a propensity score. And one sees this in other work on propensity score methods. So I'm not claiming that this is something, terribly unusual. In fact, it's kind of terribly common, I would say, in the propensity score literature, but it was news to me, and I still think of it as a kind of interesting and kind of cautionary tale when it comes to propensity score weighting and maybe more general things in other kinds of applications. Yeah, so I can say just a little bit more about this. This is I've done some additional simulations with models of this sort in regression just to sort of see how these things work in regression settings. This is clearly a situation where you probably don't want to think about quantile regression. It's kind of classical IID error model. You probably can't see very well the distinction between closed circles and open circles here, but open circles, if you can see them, are the censored observations. In the first panel, they're all on a fixed level, and in the second panel, there's random censoring. And so they're kind of scattered around in the plot. And now you can calculate bias, mean absolute error, root mean squared error for each of the estimators. And what you see from this is that Portnoy and Peng and Wang do quite comparably well. Powell estimator does a little bit worse, but maybe not as badly as the previous asymptotics would have suggested it does. And this GMLE is actually computing, since we know the distribution here, what the MLE would be if you just estimated the model using the correct likelihood. It does very well, but it's obviously assuming that we know quite a lot more than we typically would know. And the results for this is the same kind of model, but now we have variable censoring, that is random censoring, and I guess the results of the table are very similar. Then we have the same kind of configuration, except we have nonlinear quantile regression, or nonlinear conditional quantile functions. Again, it's fixed censoring and random censoring in the two panels. And now we have more things to compare. There's Portnoy L, Portnoy Q. Portnoy L means we're assuming linearity when we really shouldn't be, because everything is quadratic. Portnoy Q is assuming that we know that we should be estimating quadratics, and correspondingly for Peng and Wang, L and Q, and Powell, assuming that we know the thing is quadratic, and again the GMLE. But the GMLE is assuming that we have an IID error model, and the GMLE, because it doesn't know that we have this kind of heteroscedasticity effect, is kind of a disaster. And the other methods are reasonably competitive, I would say, and in fact, in some cases, Powell looks like it's actually doing quite a bit better in small samples than some of the other estimators, but once the sample size gets to 400 or 1,000, the Peng and Wang and Portnoy estimators are doing quite a lot better. So I think really what this means to me is that it's advantageous, at least in the random censoring cases, to think seriously about these new methods. They're relatively easily implemented in R, and like other quantile regression methods, they're really able to detect a wider variety of phenomena than the standard survival analysis models in some cases. This is the variable censoring, but these tables are very similar. So I think I don't need to talk too much about that. I can say just a little bit in the way of conclusion. I think this is going to end a little bit earlier than planned, but that's good if we could encourage more questions. There are some scatter plots that I maybe should have shown looking at the difference between the Portnoy and the Peng and Wang estimator, which are pretty convincing in terms of as sample size increases, the two procedures are delivering very, very strongly correlated results. And in simple cases where we can do asymptotics, it's possible to see that they should deliver exactly the same behavior. As I said, the martingale representation makes the Peng and Wang approach a little bit more straightforward. I should say maybe that Wang has a paper on his own in a relatively recent annals, I think last year, providing a little bit more detail on the kind of general approach that they outlined in this previous JASA paper. I have to say that I'm still very fond of this Powell estimator. I mean, I suppose partly it's just nostalgia for those days in which quantile regression was something kind of off in left field somewhere in econometrics, and not very many people were paying attention to it, but Jim Powell was paying attention even as a graduate student, and this approach that he suggested actually has been very fruitful in a lot of other contexts in econometrics. And if one looks now, even in light of more recent developments, this estimator is still performing, I think, quite well. And I guess the usual final comment would be that the idea here is obviously to try to provide a way to look at duration data that's a bit more flexible than the standard IID error kinds of models that one sees in the classical survival analysis literature. It was a huge impediment to doing anything more with this until we had ways to think about handling censoring. There are still many questions about how to handle censoring of various types that haven't been handled in the ways that we've done here. There's interval censoring is a quite important thing in a lot of biomedical examples, and also in quite a few econometric examples too. And that's something that we're only beginning to be able to deal with. Competing risk models is, I guess, the other major area where I think there is hope of doing things, but multivariate quantile modeling and estimation is something that is still quite controversial. We can't quite come to some agreement on what a multivariate median is, so it's a little bit hard to imagine having a very good general discussion of what multivariate quantile should look like in general settings, but there are certainly situations in which we've made some progress in that front. I mentioned a couple of papers in the morning talk. There's a nice recent paper by Marc Alain, David Pendevin from Brussels. Another quite nice thing by Ivan Misra and a student of his in Edmonton. And there's a quite neat paper in JASA by Ying Wei, one of the co-authors on this, the paper that I mentioned on growth curves. She wanted to do bivariate growth curves for both weight and height, and this is quite an important thing for pediatricians, and she has quite a nice and relatively simple strategy for estimating contours for weight and height as a function of age and as a function of other covariates. And this is an approach that could be easily adapted in econometric applications, I think. It requires some constraints on the form of the multivariate distribution. You really need to have convex contour sets in order to think about this working well, but in the cases where that seems an appropriate assumption, her approach is actually quite attractive. And in effect, you're just doing quantile regression in various directions, and in bivariate settings, this is quite a feasible thing. In very high dimensions with multivariate data, it may be quite a difficult thing, but a lot of competing risk models are really competing risk over only 2 or 3 alternatives, and in those settings, it seems quite plausible. I think this is the last slide and just gives a few references to this topic. And with that, I think I'll stop. The small table or that one?
After? That one?
Yeah.
That one. So what do we take from this? Comparing the different estimators, the bias seems to be the smallest for the Powell estimator, is that right?
My sense is that the bias is really pretty negligible compared to variance for all of the methods here, because
the bias is coming from the censoring, right?
Well, yeah, but the bias. I mean, there is no asymptotic bias, the bias is sort of an order of magnitude smaller than root mean squared error or mean absolute error. And so, normally you would sort of think, well, maybe we could do something to do some bias variance trade-off here, but in fact it doesn't seem like that's really necessary in a situation where there's an order of magnitude difference between the two things. I guess my main kind of interpretive message of this would be that we need to pay closer attention to the variability. And the variability it seems to be giving a slight edge to the random censoring, Portnoy-Peng-Wang thing, but you can see that Powell is really right in there. The only thing that I might add about the Powell estimator is that there are some computational difficulties, and if you really are insistent on getting the maximum for the Powell criterion function, you can sometimes find that it is doing something very strange. And so we've definitely encountered situations where mean squared error is really blown up by one or two bad realizations in a Monte Carlo experiment. That's part of the reason why I'm doing mean absolute error. I mean, I'm kind of a mean absolute error sort of person anyway, I suppose, but the need for doing both things was just sort of to make sure that we weren't distorting the comparison based on a few bad realizations. The Powell estimator can occasionally do things that are quite strange, especially if you do global optimization. But the Fitzenberger approach to doing the optimization is something like simplex, and so you're sort of starting from some kind of reasonable position and iterating towards a local optimum. And that doesn't usually produce anything too crazy, whereas occasionally doing every possible combinatorial search kind of thing finds something that is pretty crazy, but really is the minimizer, the objective function, and that's kind of disturbing when it happens. If you look at my package, this R package, there is an option for doing global optimization for Powell. I don't really recommend it for that reason, and because it also, if in a problem of decent size, it actually can be quite slow. So it may get the right answer, but it has maybe poor performance, and it's slow, so that's not good.
So I wonder how you would handle time variable in this context. Regressions which are during the duration of the spell. So is there a way forward to do this in the quantum regression framework?
This is a question that I always dread, but I have an easy answer for it today, actually, because Blaise Mely, I don't know if he's here yet, but he's giving a talk about this tomorrow, which I'm actually quite eager to hear. My standard answer is I don't really have any idea. And the reason is that there's something quite natural about using time-varying covariates in a setting where you're estimating models based on hazard function, because you have instantaneous kind of impact of the time-varying covariates up to time t on hazard at time t. But the quantile regression thing is looking at the survival distribution, and therefore you have time-varying covariates, and now somehow it's supposed to be looking back on what happened, in low quantiles of the survival distribution, and that seems like a kind of fundamental difficulty. But we'll see tomorrow.
When you do this level analysis, normally you have a small sample size. You don't have about 2 million observations.
Yeah.
And also, when you do the analysis, sometimes you want to do the identifications to find the good instrumental variables. So can you please give some comments on using this quantile regressions, the advantage of using quantile regressions regarding the size good instrument?
Yeah, I mean, as a general principle, I'm always hoping to get large data sets, I would say. I mean, I think, you know, a part of the rationale for conventional survival models and conventional least squares analysis in a lot of small samples, I mean, small data sets, is that, you know, there's nothing more that we can hope to learn than something like the central tendency of the response as a function of covariates. And so, you know, it's very unrealistic to think that we can learn a lot about, you know, extreme tail behavior unless we're in this kind of, you know, medfly sort of situation where we have an embarrassment of riches in the data side. And I guess in situations, as you might often expect in econometrics, where you have survival models with endogeneity, the existing procedures certainly would expect you to have richer data sets than maybe one would typically have in a lot of experimental settings, certainly. But at the same time, you know, we see more and more larger data sets in these kinds of applications. Maybe not so much in the clinical trial domain, but in economic experiments of various sorts. It's increasingly common in, say, school evaluation program type evaluation that we're seeing quite large data sets. And in those cases, I think there's really a strong, I was going to say obligation, but that maybe is too strong a word, but you know, some kind of rationale for a more extensive analysis that's a little bit more flexible than standard Cox model stuff. But you know, these things are always you know, subject to the really detail of the data set, I think. I mean, I'm quite suspicious of quantile regression analysis in general in situations where the sample size isn't, you know, a few hundred. And so, you know, for some people that may rule out these methods entirely. They just never see data sets that big. But a lot of people are seeing data sets that are, you know, much bigger than my 1.2 million. And for those kinds of people, then there's probably a lot more that can be learned than just by estimating mean models.
You didn't apply the methods to your medfly sample. I mean, it would be nice to see what those estimates are any different across the.
Yeah, well, you mean I mean, yeah, maybe I could have done that by doing some kind of, you know, artificial censoring, but without any censoring, then, you know, then all of these methods reduce back to the original simple quantile regression things.
Really uncensored.
Right. Right. I mean, you know, they all died after less than a year, and they were all swept out of the bottom of the cage. And every time I think about, you know, these guys who are actually looking at with their microscopes to decide what sex these flies were, I kind of cringe. But it's you know, it was quite a data collection effort.